It's loading. There we go. All right. Good afternoon. Hope you guys are having an awesome day. Let's talk about the sponges. So porifera comes from the Latin, which means pore bearing, and basically it refers to all those tiny openings, and you can see those in the picture. The word pore just means opening, like you have pores on your face. So that's where porifera comes from. So their level of organization in sponges stops at cells. These are the only sponges that do not have tissues. So they are the least complex animal. And a sponge is really just a colony or collection of a bunch of single cells attached to each other, like we talked about when we watched the porifera video. We also talked about how they exhibit cell recognition. So we can actually separate those cells from one another, and over time, they'll begin to communicate, talk to each other, and find their way back into the original spot that they were in that sponge colony organism. You can't see me, but I'm doing air quotes. Okay, so we watched how sponges are filter feeders. Since every individual cell is taking in nutrients, they can't eat anything larger than what one cell would eat. So everything has to be microscopic. And there's a lot of microscopic particles floating out in the ocean, things like bacteria, algae, protozoans, which are single-celled organisms. And they ingest, well, they do ingest, but through endocytosis, through the cell membrane. Newly developing sponges are mobile. The adults are immobile, which we call sessile. And so it's for this reason that sponges were originally classified as plants, because they didn't move. Now, those mobile sponges, they're basically just at the mercy of the ocean currents. There's no brain, so they can't consciously go somewhere. They just float along until they hit a substrate and attach to something, and then they'll continue to grow. Sponges are very diverse, and they're based on their support system. There are two types of support system. You can have spongin, which is a very durable protein fiber. It's also flexible, so it allows some movement. And you can also have spicules, which are tiny needles of two different chemical components, silica or calcium carbonate. There are a few species of sponges that have both of these in their support system, just in case you're ever on Jeopardy someday, but we're just going to differentiate between the spongin and spicules. These three classes that we're going to talk about have either or, not both. Okay, so we are in the kingdom Animalia, and we are in the phylum Porifera, so we're only talking about sponges. Within the phylum Porifera, we can divide them into three main groups or classes. The first one is the calcareous sponge. These spicules are made of calcium carbonate. You can remember this because look, Cal, calcium, Car, carbonate. So you'll always know that their spicules are made of CaCO3. They live in the shallow tropical ocean waters. If you've ever gone snorkeling or scuba diving, you may have seen these. The second class are the glass sponges. Their spicules are made of silica. They're found in the deep, deep ocean. Notice the color difference, and especially in the cold waters of the Antarctic. The third class are demisponges. Their support system is spongin. Basically, most sponges are in this group, including the few freshwater species that we have. Okay, now we're going to start sponge anatomy, and you're going to need your diagram here in just a little minute. So if you haven't picked that up, go grab it. All right, so sponges consist of two layers. In between those two layers is a non-living jelly substance called the mesoglea. The outer layer of the sponge is its epidermis. That's the part that touches the ocean. The inner layer is the gastrodermis. That's the layer that lines the central cavity. And in between there is the mesoglea. So on your diagram, sponges are supported, in this case, by spicules. So what you're going to do is you're going to color your spicules brown. They're all the little things that are sticking out that look like ninja throwing stars. They're hardened particles, you know that. They also stick out of the epidermis, help hold it together. The epithelial cells form the epidermis. You're going to color those pink. They cover that outer body and form the epidermis, which I just said. Okay. The central cavity is lined with collar cells. In the video, they called them chanocytes. That's also known as, I like to say collar cells. I told you guys that. So all those little cells that look like bombs with eyeballs on them, those are the collar cells. And each one of those little fuses on that bomb is a flagellum. And it's the rotation of those flagellum, or flagella, plural, that circulate water through the sponge. All right, next we have already talked about the collar cells. Okay, so they rotate their flagella. When they do that, they're pulling water in through the in-current pores, which are light blue. And you'll notice if you look on there, so this is a cross section, they look like the little donuts. So all of them are like these little tunnels that traverse the epidermis, the mesoglia, and the gastrodermis into that central cavity. Water then will be sucked in, and then it travels out the top like we saw in the video with the dye particles, and it travels out what we call that top part, the osculum. Okay, the amoebocytes are the things that look like little fried eggs in the mesoglia. We're going to color those green. They travel around distributing food and oxygen to the outer cells as needed. So they're little helpers. We appreciate them. And then when you're all done, this is how it should look, except that I apparently didn't color the spicules before I put this in the presentation, but oh well. There you go, the end. Have fun making your flip book.